Hello, I'm Darren Woodside, the Vice President for Research here at the Texas Heart Institute, and welcome to In the Studio with our Grand Round speaker today, Dr. Wang. Dr. Wang is a professor of medicine from the University of South Florida. He's also the director of the Center for Regenerative Medicine. Dr. Wang today provided a really interesting seminar on how certain genes in our body help to develop the heart and help to have the heart function under normal situations. Welcome, Dr. Wang, and thank you for a very nice talk today.
Thank you so much for this opportunity.
Your career has been pioneering the defining the mechanisms of how kind of a unique regulatory pathway occurs in heart development and heart function. It's kind of interesting that the Human Genome Project that began over decades ago tried to identify all the genes in the human body. And I think it surprised a lot of scientists when there was only like 20,000 or so, right?
Right,
or even less than that were identified.
Yeah.
But it turns out the genes in our body actually encode for a lot of other things that aren't just proteins, right? So if everyone can remember back to Biology 101, where you have genes that are transcribed into messages, which are then translated into all the proteins in our body. Only a small percentage of those genes actually make proteins. But a lot of the other genes make these other things that you've devoted your career to studying in heart development and function. I wonder, they're called short RNAs. I wonder if you could actually just describe in very general terms what these short RNAs are and how they function.
Yeah. So actually, just like you mentioned that our genome, really a lot of the DNA elements were encoded for those unknown stuff. Eventually, they were made into those very small molecules, we call them molecules, that don't encode for protein, but what they were used for is sometimes regulated the process of the gene expressing. So now we learn more and more of those importance of those small molecules. Some of them are very so-called tissue specific. For example, some of them only exist in the heart, in the cardiomyocytes, the muscle cells. Some of them are very specific, for example, in neuron or in liver. And they appear to be very critical for some of the very specific processes, including in the heart disease scenario. If there is some stress condition, those molecules were either induced or repressed. Then what they come up with is, hey, I detect there is some changes. I got to come to do my job. What they come out with is very specific and control, say, this gene pathway need to be turning on to really deal with the stress. Isn't that fascinating?
It is, yeah. These are small pieces of genetic material that aren't part of your genome itself, but they're expressed to regulate other genes that are important in many physiological processes.
Exactly. That's a really evolutionized how we think about the traditional genetic terms, as you mentioned, the gene, transcripts, and proteins. Now there are some of many of those genome components were made as not a protein at all, but they are so important for some of the biological process or function in our heart, in our bodies.
Yeah. For the budding of scientists out there, how do you get interested in an area that's kind of it's outside of the fundamental processes of biology, really? When it's something so new, at the time, no one really understood the complexity of these small genetic materials that could regulate so many different things. How do you fall into that as a scientist?
Wow, that's a great question. Actually, you just reminded me when I started this work when I was just a junior faculty. I was trying to find my own sort of like a path to say what kind of question I am interested. So I start to go to talks and read papers, and I was like, wow, there is some finding that many of those again, our genome were unknown. There are so many new stuff. Then I thought that they got to be something. They got to do something. So we start to read and think about it. And another thing I was trying to say is, when we were in that situation, actually, we have to almost like invent many of the wheels, because we could not use the conventional method to detect them or to show they're expressing. So we almost have to, again, try to use a new method to show them. And that was a lot of fun. Of course, it's always a lot of challenges, but we are so happy that we and many other colleagues and we learn new stuff. I think that's, in some sense, that's how a scientist is supposed to do.
Right.
New discoveries and trying to solve the question and problems.
I think the biggest component of a scientist's desire to do the work is really curiosity, right?
Yeah.
Here you're faced with a situation where most of our genetic code doesn't make protein. So what does it do? And when you start interrogating different lines of questioning, the results lead you down a pathway that just keeps providing more questions and more questions.
Yeah.
A scientist's work is never done, really.
Yeah. And I also want to say, I think when I visit Texas Heart Institute, I know here, I mean, scientists or the service people have done such a fantastic job. The translational need of this really translate what we learn about into potential therapeutic target or clinical treatment was so important. So in the case of microRNAs, small RNAs, we studied. Nowadays, we understood them a little bit more. So our next goal is really trying to see whether we could put those into, again, the drug development or potential treatment of those patients with heart diseases. I think that is ultimate goal for us to really help human beings treat the patient. I know THI has done a fantastic job for the translation, and I just am looking forward to hear more about the exciting thing here too.
Yes, thank you for that. That it does, it rings true in all the work that we do here. We try to translate a lot of the basic fundamental research that occurs, for instance, in your lab and other labs that we collaborate with, translate that into new treatments or new devices that can help patients with heart failure or ischemic heart disease and things like that. Yeah. The beauty of the Texas Heart Institute is that we can work in lockstep with clinicians that see patients every day and they identify the unmet needs that they see. They can work directly with the scientists and clinician scientists here at the Institute to help develop new strategies to treat those patients. From the perspective of developing a new maybe drug to treat patients with cardiovascular diseases, when you're targeting small genetic materials that can have broad specificities with things that they can affect a lot of different things, right? What are some of the issues you foresee in the future trying to develop new therapies that can affect a lot of different things? Because the drug industry likes to have one target and one drug, right? Yeah. And you inhibit those things and you see an effect. When you talk about regulating some of these small genetic materials that are in our bodies that can have a lot of different downstream effects, what are some of the regulatory issues that you might see that would pop up that might actually be very difficult to overcome?
Right, yeah. That's, again, always coming up as such an important question. So I think there are several points. One is, after we have learned some of the smaller moleculars, as we discussed about it in the Grand Round, we start to understand how specificity of those molecular really is built up. That will be very beneficial for us to look into their druggable targets, because the specificity is unique, is important. So when we learn more about this, then we get more hands-on on how we can control it. And the other thing I'm thinking about is our heart is such a critical organ. So there are multiple bioengineer or therapeutic like a gene therapy type of approach is trying to specifically target some of the cardiac muscles. I think that will help us to more specifically deal with our cardiac disease instead of a systematic whole body. And then the other thing I was thinking about is because of the so essential functioning of the small molecules in the heart, they probably will play in a concert of many protein coding genes, proteins. So if we can use the small molecular we study with the carrier of the drug or inhibitor, then they can specifically target some of the proteins, or like, for example, the contractile proteins or inflammatory response proteins in the heart. That may help to develop a new direction of therapy, actually.
Well, thank you.
Yeah.
Thank you for sharing your time with us today at the Texas Heart Institute. Thank you for your grand rounds, and keep up the excellent fundamental research so we can drive that translational development program.
Great. Thank you for the opportunity. Yeah.
Thank you.
It's been so fun.